Yeah, good afternoon, everybody, and welcome to our panel. We will talk today about identifying, progressing, and financing innovation. For the purpose of this discussion, we kept the term innovation somewhat broader. So we are not talking only about scientific innovation, which is, of course, the basis for attracting investors and creating a company, strong science, breakthrough science, ideally. But we talk also about innovation in translation, in innovation to really demonstrate clinical proof of concept, which is particularly important in the field of oncology, as the predictability of preclinical models is limited, and many partners want to see clinical data. So it's an important aspect to be creative and innovative, how you're going to go to characterize your assets for efficacy. And lastly, also about then the innovation that you need to develop successfully to the market, right? So, particularly oncology, you're dealing with multiple indications, you're dealing with combination treatments. This can become very quickly extremely expensive, and you have to be smart with your development strategy. And we talk about innovative concepts in this field. So that's the topic for today. I want to introduce my co-chair Esteban, who takes over the introduction of the participant and the companies.
Thank you very much, Andreas. I'm Esteban Pombo. Worked with TargImmune Therapeutics for the last 8 years, where we developed a targeted nanoparticle delivery of double-stranded RNA that we can provide systemically to solid tumors and metastases, inducing apoptosis and a strong immune response. So very good effects in solid tumors. And that has brought my interest in immuno-oncology over the last 8 years quite significantly. It is really a pleasure for me today to have our celebrated panelists to explore, as Andreas said, all different aspects of innovation. How science, of course, is the basis, but how you need to overcome challenges in all different aspects of company creation, from funding, fundraising, preclinical project design, clinical data, and interaction with shareholders. So I believe that in the course of today, we will have very specific insights into how each of the companies that we have represented here have overcome challenges by being creative and innovative in taking these technologies into what will be the therapy of cancer in the future. So if I invite Apollon to give a brief introduction, please, Apollon.
Yeah, thank you, Esteban. Thank you, Esteban. Hello, everybody. My name is Apollon Papadimitriou. I'm the CEO of iOmx Therapeutics. We are a Munich-based company, clinical stage company, focused on targeting novel immune escape pathways, meaning we are fully into immuno-oncology. To my personal background, I'm a biochemist by training. Before I started with iOmx, I've been for more than 20 years with a company called Roche. Now, with respect to iOmx, our specialty is to identify new targets and drug them with multi-specific therapeutics, specifically antibodies that tackle more than one pathway. And I believe this is maybe a path forward into making immuno-oncology more powerful, more potently, getting away from combination treatment only.
Thank you very much, Apollon. I would like to invite Erik for the next presentation.
Thank you. So good afternoon, everybody. So I'm Erik Halioua, the executive chairman of the French company Ervimmune. Ervimmune is developing different modalities, including cancer vaccine, targeting specific class of antigens called endogenous retrovirus, human endogenous retrovirus, that are specifically expressed in tumor and not in healthy cells. The lead asset of the company that is going to enter a clinical trial beginning of next year is in triple-negative breast cancer and ovarian cancer. We recently raised a Series A of 17 million euros. And I joined in January at the same time that the Series A, the round is still open for new investors. So new investors are welcome for new money. And so I'm very pleased to be in this panel to be able to discuss about innovations in the field. Thank you.
Thank you very much, Erik. Now it's my pleasure to invite Erik to the presentation.
Yeah, it's a common name, so a pleasure all to be here. Thank you. Erik van den Berg, CEO of Memo Therapeutics. We have a phase 3 ready asset outside of oncology, which is an antibody that addresses the BK virus in transplantation. But we do also, critically for this panel, address oncology by identifying novel targets derived from antibodies that are produced by B cells residing in the tumors. So we start with resection of tumors and then identify what antibodies are expressed and what targets are they hitting. Because we're a firm believer that we need good and additional targets in the oncology space. We cannot continue, of course, to focus on CD20 and BCMA and other populated targets. Maybe also relevant for the panel, I am chairperson of STEP Pharma, both in T-cell lymphomas and in clinical trials in solid tumors, addressing CTPS2 deleted cancers. And originally being a co-founder and executive chairman of Lava Therapeutics. It was a T-cell engager company focusing on gamma delta T-cells. Pleasure to have joined the panel and have a good discussion indeed about innovation, which is in the end bringing products or services to market to use. And there's a lot coming in that development besides science. In clinical development was already highlighted. And of course, how do we fund this all?
Thank you very much, Erik. I would like to invite Luca to present his company and himself.
Yeah, thanks. Thanks a lot for the invitation and for being in this panel. So my name is Luca Cassetta. I'm the founder and the VP of Immunology of a company called Macomics. So Macomics is a preclinical company which is focusing on targeting macrophages in different indications, mostly oncology, but we're also looking in CNS and other diseases like fibrosis. We have two programs, one in IND studies and the other one in pre-IND studies. And when we are discussing about innovation, you will also see that we need to evolve a platform for gene editing of macrophages, which supported really the target validation, but also the drug discovery programs that we are running in our company. In terms of myself, I'm mostly an academic by origin, so I spent all my life in academia, and then I decided to jump into this magic world of biotech 5 years ago. I'm a macrophage immunologist by training, so I work in virology and then in oncology for many, many years. So I will probably bring the perspective of an academic moving to biotech and what means innovation for me as well. So probably this will be different from the type of experience that you had. So thanks.
Thank you very much, Luca. Romana, please.
Yes, so my name is Romana Gugenberger. I'm on the board of invIOs. invIOs is a Vienna-based biotech company where we develop cell therapy, but as also small molecules. So the idea for us in cell therapy was that there is a need to do so, and our focus is strongly on manufacturing. How can we reduce the costs? And the idea is to have a point-of-care manufacturing. Therefore, we developed our EPIC platform, and on this platform we currently have 4 programs running in different stages between discovery or the very early preclinical, but also we have completed phase 1 program here running using different kind of cells, using different kind of modifications of the cells. And based on that experience we gained here, we also did a phenotypic screen for looking for small molecules that also have an immune cell activation as an idea behind that. And here also we have more programs running. Our most advanced program is in IND enabling studies. Yes, to my person, I'm a molecular biologist by training, and after several years academic research, now I switched to the biotech world now more than 15 years ago. And was also on the board of the mother company of invIOs. This was Apollon Biologics, where we brought also a product to the market for neuroblastoma. Yes, and finally, Apollon was sold not 1.5 years ago to a big US company, and yeah, happy to join here.
Yeah, thanks everybody. Maybe quickly to me. So I'm Andreas Wallnoefer. I spent quite some time at Hoffmann La Roche, was 14 years in their R&D leadership team, did things like head of clinical research, but also led their development for some time and was involved in several drugs that reached the market. I left Roche after Genentech, then was an investor, had the privilege to be involved in some successful company startups, exits after the last one, of the company that we sold to Merck in 24. I stepped out of investment, became an independent board member, and currently are working on a few very interesting early companies. My additional role is, and you see Clavist. Clavist is the early investment arm of Temasek. Temasek is the Singapore state fund, and I'm working with them particularly on developing innovation in Asia, or maybe interesting in this context, building the bridge for European companies to develop in Asia together with the Temasek company. So having said enough here, we want to launch the discussion. So the first question, remember we said the theme is innovation. If you think about the first step, which is essentially innovation as the basis of the company, the innovation that actually really is making the company differentiated, attracted for investors. So let me throw that question in the round, particularly for the companies that are involved in research and preclinical stage. How do you profile, how do you create and differentiate your innovation that basically you stick out of the big crowd? Who wants to go first?
Yeah, I can go first if you want. Yeah, that's that's not an easy journey, right? Because it's crowded out there. I guess I can bring my own experience on when we founded the company. Of course, we're talking about macrophages, myeloid. So if you think about immuno-oncology, everything was really T-cell based initially. So we had to really go and convince people, first of all, that what we found in academia was actually true. All these papers that we published were making sense and was not just an academic exercise. So that was probably the first challenge, to show innovation on how to find a way to target macrophages in a smart way so that we can actually have an impact. And that was really, so I think it goes in stages. First of all, convincing people that you have a target that really makes sense. And over there, of course, if you think about the nature of macrophages and the nature of immune cells that we're targeting, we're talking about redundancy, so really showing that the signal is strong, and then we can actually target it with a molecule. But then the challenges in innovation are not only about the targets, is how you, as we discussed, about how you show the signal and how you convince people that your signal is true. And in this case, of course, you're still all the models that we had to use and face were all based on T cells. So we had to really navigate innovation in terms of creating new models, trying to make sure the macrophages were really fully represented at the preclinical level. We're talking about in vivo. We're talking about reproducing what we see in the tumor ecosystem in vitro, so setting up new assays. And that's why that led to the creation of Enigma, this platform, using iPSC-derived macrophages that we can gene edit so that we can reproduce the tumor microenvironment in vitro. And that was really an innovation step that we had to go through. So it doesn't stop in identification of a target. It's also, building up a structure that really allows us to drive this target through the preclinical stage and be convincing with a signal that we are actually translating something into potential clinical signal in the future. So it's a lot to build up, because of course we are building up a new model and a new kind of system.
And if you take it to I think Luca, so it's if I can summarize, maybe also it's about attractiveness then, of making your platform attractive or your technology, and it's about credibility. So if you then put the science in motion and of course, we all need fuel, which means money. What can be then in convincing elements that you need to think through, so that credibility and attractiveness comes back, of course, in the science, but also in the team that is employed. Then the plan that you develop on how you can create value of that to a potential exit is important. That the plan also needs to be credible. And of course, you have to address an attractive market where you can differentiate in. Those are important elements to work with. Of course, IP is helpful as well, so that you have a position where you can be dominant in. And I think on those 5 elements, it's important to show clearly, of course, and make it credible to investors that this is a unique opportunity to invest in.
But Erik, what you're saying is, and I think that's a trap that you see for particularly very academic startup CEOs. The idea that the great science, the Nature paper, that will do the job alone, right? So what you're saying, essentially, this is great, but then actually even in this early phase, when you build the company, you need to amalgamate that with the biotech experience about how the company could look like and the product and potentially the exit, right? So I think that's a critical stage that probably separates the winners from the losers in a way, paradoxically independent of the science. The science can be super good. If you put the wrong people on it, you're not going anywhere, right? I mean, Apollon, what you're nodding.
Yeah, no, I think you summarized this very well, and I can speak for iOmx. You know, the iOmx started as a company that hunts for or identifies new targets beyond PD-1, PD-L1, right? New immune checkpoints. That was the story. That was, the idea at the very beginning. The vision for the company was, we will identify the next PD-L1 on tumor cells, and we will drug this and generate value out of this. This was very exciting, and this was very academic. So there was a high throughput technology that allows you to screen individual genes. But very rapidly, already in year 1 of after the foundation of the company, the question was not about the targets. The question was about, do we have products, right? And this is a completely different skill set that you need in your company, to have the robust science, to validate a target, to confirm a target, but then to convert this knowledge around the target into a product. This is something completely different. And this is what we realized in our hands, that it needs completely different skill sets, and it needs speed, innovation in how would I generate this clinical signal as quickly as possible under limited financial flexibility that every one of us has, right? So I think every company has to go through a transition from a discovery type, very visionary thing into something very product relevant, very development focused. And the faster you can do this, the better you are off.
I mean, from my observation, how do you deal with, from my observation, what you see is that there are now several very large funds that have to deploy capital. And they typically tell you, this is fantastic innovation. Why don't you come back once you are in clinics? How do you actually motivate people to finance the first part, right? And how do you do this to keep the momentum and the speed, right?
It's a tricky part, as you explained. Before having clinical trial, clinical results, sometimes you can do some academic clinical trials to help to have some of this data. But if you don't have any data in human, it becomes very difficult now to go out from the crowd. But definitely you need to start. You need to start to have a product. You need to have a plan and to be ready to start a clinical trial. Because if you go too quickly in clinical trial without the right CMC, without the right product, the right coverage, right analysis of the situation, and the right plan, you're going to fail. So it's a tricky part, and I think it's where you need, as you said, experience and the right leadership. To have the team and the right people in the team to be able to develop the plan, to be credible, as the other Erik said, to the investors. and after to be genius in terms of investment. In some country, you can have a lot of grants, you can maximize, the sources of funding, and if you know where to go. In the case of Ervimmune, they get the EIC accelerator grants that help a lot. It provided additional credibility, and when the investor came, and they succeed to convince investor to finance the clinical trial that will start next year. They said, okay, we need to reinforce the team, and I joined, and we are going to have some very senior CMC people, etc. So I would say the investor can help as well, if you have the right investor, to provide the right tools to be, we hope, a successful company. But what you said is very important. It's very tricky to pass from a discovery company to a product development company, and it's not straightforward. So you need a bit of experience. That's what I would say. Yeah.
Yeah, I mean, which brings us to the next opportunity and challenge, right? So if I talk now to my pharma colleagues, they typically say this is fantastic, but really to believe in it, we need to have some clinical data, right? So now, having said that, the debate typically is what is convincing clinical data, particularly in oncology. There, not rarely you start your first trials in very advanced patients. You need to be very careful about who is really participating in your trials, because people are looking for these famous complete or partial responders, and how do you find them, right? Romana, you are in clinical. What is your experience there? How can you be smart and get the package that really motivates people or pharma to really seriously look into your company?
So we made exactly that observation. So we did a clinical phase 1 using our cell therapy, which was a very difficult patient population, especially if you're in phase 1, you get really highly advanced patient. We are focusing on solid cancers, so we did it in a basket, so it was really a mixture of the patients, and the patients received between 5 and 11 treatment lines before. So especially as a cell, when we are working with autologous cells, so we need some material of the patients with which we can work. Since our therapy is not only focusing on T cells, since the checkpoint with that we are working also activates the cells from the innate immune system. So there is also not only to have enough T cells. And what we found out that the T cell is not the trigger even to show clinical response, especially in that patient population. But it's exactly that. And then you go to an investor and say, okay, we say that we saw in this clinical trial that the classical 35 to 40% of the patients responded, but we don't see here a partial response or a complete response, because it's nearly not possible to get some signals like that. What we saw is that the patients responded. We saw changes, of course, in the biomarker. We saw changes in the immune cell distribution. So we saw that the therapy, or especially the responded, react on that. We saw that the patients had a stable disease, which were even not before. So they came all with progressive disease in the clinical trial. But this looks like if not enough to convince investors or someone else. So it's really a problem. So then comes the comparison to what was in the clinical development for 5 years ago, 10 years ago. And then you are really in a strange discussion, because we still have not the same patients yet. So for the patients, a stabilization of 2 years might be a great thing, because they have quality of life, they have no additional therapies that are ongoing. But for a real clinical signal, it is not enough to convince any investor.
One important new element, or relatively new element, in data analysis, prediction, and modeling is the so-called artificial intelligence variety of tools that are available. And there's applications to target discovery, there's applications to clinical development. What do you see is the potential impact so far in the way you have applied these tools, and where do you see these tools can help you, for example, generate data packages that are convincing to stakeholders like investors, for example?
Well, at Memo, at least, we're not yet, say, integrating it into our antibody discovery process, although we think about that indeed, and we're trying to develop it. Where we have found most use of AI, besides, say, the general stuff, is to actually build programs to analyze data, because it's very good in coding and generating code. So that's really helpful because it provides additional insight and data that can be very complex to analyze via more traditional statistical methods. So that's one. And the second is, which might be useful for many of the biotechs here as well, it's very good in generating answers to the many due diligence questions that you get. Because that can occupy quite a bit of your team, right? If you're in multiple due diligence together, and if you have a repository of all your Q&As that you have done before, it can build the answers relatively quickly for you. So it's maybe a bit off the innovation track, but it's still very practical.
It's definitely labor savings. So, on the one hand, the labor saving on some sort of mundane activities that would demand a lot of expert time to actually come up with a relatively trivial result is now liberated for some really creative, in-depth thought process.
Yeah.
Somebody else has a similar application?
So we tried now also to find a partner, an AI partner for development of the small molecule. As I said, we have developed one which is now before IND, and we have out from this phenotypic screen additional substance, different substance classes that would be of interest. And therefore we thought for the medchem part it would be a classical AI field to support that, to accelerate and to streamline that. And since we took 3 to 4 months where we really spoke to a lot of AI companies to support exactly that approach. And it was, to be honest, a little bit disappointing because mainly there are the databases behind are rare. The main focus was on to share our data on the already developed, where we have our clinical candidate now ready, because here we need approximately 1,500 different compounds were synthesized until we came to that we have a clinical candidate. And this was the main focus of the company, to really have access to our data, how successful we are to follow on. And then, of course, we discussed this with experts, and they said also that their experience is that if you do that AI-driven, you need not 1,500, you need between 3,000 and 5,000 compounds that has to be synthesized until you reach the goal of a clinical candidate. And so also maybe this, where we really thought that would be the perfect match for AI, because I think there is nothing special here to add which compounds here. But yes, this was our experience.
So really the challenge for every computational method that we have used in medicinal chemistry is the quality of the input data that creates the quality model that is then predictive for the test case. And it's clear that in some cases, you just don't have enough data to create a predictive model in spite of all these smart ways of analyzing data and being generative AI. But based on a training set, and the quality of the training set is essential. Has anybody applied this to clinical trial design or Bayesian type of analytics, or is that something that you see potential for patient biomarker responses, genomic studies, or something that you can apply to get robust clinical signals?
So maybe not exactly what you are saying, but for example, we have been using for one of our assets, which is currently in phase 2, we had a nice preclinical package, right? A translational package that hinted towards certain indications, right? Based on in vitro data, based on in vivo data, based on PDX data, ex vivo data, etc. But, still we wanted to refine this model, right? To predict what is the best indication for this product. And we were using AI to extract data from in-house databases, but also from public databases, and hint us towards the right indication. And what we did was we developed a predictive gene signature, a complex expression pattern in a way, based on RNA expression levels, which was derived from databases. And in vitro, this was super in predicting sensitivity to our drug, right? And this is something we have implemented into our clinical study. So we are collecting samples during phase 2, and we will do a retrospective analysis, but we were not, daring to use this in a prospective fashion and say we will only test those patients that are positive, right? But let's see. This is something where I see great value if you have a complex mixture of data to extract the right, signature or identify a marker in a complex data set.
I mean, that's a very interesting observation, right? I mean, where we are with the discussion is being innovative for confirming a clinical signal, creating enough evidence of translation, right? So I think you probably have to go beyond just the simple tolerability testing to find those signals. And what you describe, Apollon, is increases the confidence, right, in the way you could do that, right? But it's a very critical phase that you have to overcome, because typically in the next phase, and you have been successful, Apollon, is that the next phase, if you really have to go to phase 2, you talk about different tumor types, you talk about different combinations. FDA wants you to show contribution of component. So, I mean, you're talking about a total different investment ticket that needs to be justified. That's the big hurdle here. And this package, right? This package that actually creates the confidence with your investors, I think that's the key point. Erik, you were wanted to say something about that?
Yeah, not in clinical because the company did not use yet this tool, but in the case of Ervimmune, we have a database of the sequences of ERV. 8% of the genomes is based on this ERV due to old infections. And from these 400,000 sequences, 40 has been selected through a lot of in silico analysis, in vitro, in vivo analysis. And here machine learning has been used, which is the first step of AI, because behind AI you have a lot of different elements. But I believe, the information is proprietary. So we have to develop a really proprietary element to, what do you call it, a hub, specific hub just for us, and to develop the tool applied for our own technology and our own information. So as long as you can have good quality input, good quality information, and enough information, this AI system can provide interesting output, definitely in discovery, in the kind of research we're doing.
Is that effort recognized by investors and pharma, or is that more like for you increasing the efficiency?
I cannot answer for pharma. I have to test it, but it has been published. The algorithm has been explained, how we are using it, and recognized as an interesting tool to develop. So after, of course, it would be interesting to have the feedback of pharma. Yeah.
Yeah, if I can add something around machine learning, and it's probably the early stage AI, but anyway, that was also something that was really fundamental for us when we started. Because when we sequence macrophages in cancer, we also sequence the blood and monocytes. And we were able to identify gene signature that was predictive to how the patient behaves in the tissue. And of course, the application is following up the patient by taking a blood draw and isolating monocytes and looking for this gene signature and see how they respond to therapy. So of course, that's something that can predictive tell you also how they behave in clinical trials. So that absolutely. And of course, I echo what Erik said, in terms of productivity, AI is definitely making our life easier, especially if you're analyzing thousands and thousands of data sets where you're trying to combine all together, not only oncology, because I'm also a very, very big fond of comparative biology. So when I try to identify a target, I really look at different other diseases where macrophages are important and see if they behave similarly. And of course, especially in immunosuppression, for example, we compare a lot with pregnancy, where there is a lot of immune suppression going on, but it's reversible. So that we really look for that. And, that definitely helped narrowing down the genes.
Let me put another fish on the table here. I said I'm working with Temasek, which is this Asian investor, right? So what do you see? That's also not new, but I think a challenge that we have to address if you now look into how Chinese companies can process. By the time we optimize our preclinical development. They already start the clinical studies. They recruit very fast. How are we going to go to deal, particularly in oncology, with this challenge? Is it just we have to accept it, or are there ways that we actually could get back to more speed, or maybe even leveraging this opportunity to work with parties that can have access to these kind of Chinese trials? Is that something you're talking about?
Definitely a topic, yeah. But it's all of the above that you highlighted. So there are some biotechs, of course, that take the approach that you partner with a Chinese company that does the early clinical development, and then you can chime in back again, say on your own compound later on for developing in the rest of the world. But I think in the basis, something else that you see going on, especially in the Western world, is that companies stay longer in stealth mode before they actually say they attracted certain investors on board, because that's a trigger, of course, for the Chinese companies to dive in novel biology as well. So that's a practical way how to deal with it. But yeah, how can you do it as a biotech company? You have to, identify, I think, science that will remain defendable against potential copycats that can follow on relatively quickly. So in small molecule companies, like for example in Step Pharma, we ensured that we just patent the entire chemical space. And that has been a very effective barrier because we haven't seen any, say, quick followers coming afterwards. But it took a long time to get that chemical space completely patented before we went forward.
So I see creativity and innovation in potentially new models of partnering and working together with companies that are very fast, or creating barriers to competition by very, very robust processing IP. I think one other part is this process innovation in general that we have touched a little bit on, the use of certain novel tools to facilitate certain internal processes. So, China can be very fast for a number of reasons. And some of the reasons why European companies cannot be that fast may be amenable to process re-engineering using these AI and knowledge management tools in terms of facilitating interactions with regulatory authorities, creating briefing books much faster, sort of a number of elements that can accelerate the process. Do you have an example of something that can accelerate your development process in speeding things up, and maybe not as fast as China, but at least providing an approach to an accelerated, innovative development plan?
Maybe I can comment on that. So it starts with the regulatory process, right? What I'm hearing, and I think that's nothing new for the community here, Australia, right? They require very little, IND-like, regulatory processes for a first-in-human study. So this is the first thing where you can save easily 3 or 4 months, from the critical path activity if you do your first-in-man study in Australia. Then contracting, that's the stupid, believed easy things. Contracting sites, If you look into contract harmonization in Europe, how difficult it is, to contract individual sites. You have to negotiate with every single site. And here I have to mention Spain, that is really super attractive for us as, a biotech company to do a clinical study. You have harmonized contracts, etc. If you compare this with Germany, I can tell you that in Germany, the average contracting for a clinical site is 9 months. And now they are doing a big effort to reduce it to 6 months. Imagine for a biotech company, if you tell your investor, we have a program ready, but we need to bring the site under contract, and this takes 8 months. This is incredible. So these are the tiny little things, but this is where things need to improve significantly.
I think this is very important, right? So just to structure the discussion we were talking about scientific innovation, then we were talking about signal detection, clinical validation. And I think what I hear from the panel is that the innovation, how you run your program, the examples that you gave, Apollon and Romana, I think that's, if you want to be successful, you have to think about it. It's not the standard process. Again, I not want to make here advertisement, but basically what we are trying to do now with Temasek and Singapore is essentially to be a partner for European companies for Asian development. I think that Europe needs to wake up from an attitude that you want to defend, to think about what are the solutions, right? You cannot just, it's not going to go to work just to expect that you can close these parties out. We have to be part of the game.
One thing that Madame Lagarde said a couple of days ago is that they were creating a business environment in Europe where you can create a company in 3 days, and you can also wind it down in 3 days if things don't work. One thing that is really very labor intensive is company creation. It takes a long time in some places to actually build a company, and it takes even longer, and it's very expensive to wind things down. So to improve the ability of the system to create innovation, this barrier needs to be much lower. Company creation should be easy, company wind down should be easy and not terribly expensive. So that again is not directly in this innovation here. This is all company creation, and we're going to build successful companies. But the idea is those processes that are business processes, regulatory processes, should be facilitated in Europe.
Yeah, but I mean, we can't change this here. The question here is essentially how these entrepreneurs can navigate with the situation as is, right? And I think Apollon's example was very nice. I mean, basically, if Spain has a better system of site contracting, okay, let's figure this out. But you need to be aware about, I mean, it's about execution, excellence, right? So you can only hope that if people know how to do it, that the system will have to adjust. Waiting for the EU Commission to change all of this, I mean, that can take some time. So, but we need to win now, right?
Maybe, Andreas, also to add to your point on the clinical development strategy and the biology and the science behind the product. I mean, and we are talking a lot about immuno-oncology, and here, you know, the history has shown that these combination approaches did not really work out. And if it takes 5 years of clinical development and a full phase 2 study to figure out that your initial combination signal that you have generated in a phase 1 is not a true signal, I mean, that's also tough, right? That's something that you cannot blame anybody. You need to learn and adapt your own company strategy. And therefore, you know, my plaidoyer is that we should aim for single agent efficacy for these IO drugs. You know, that is, I think, a key remit within iOmx. You know, every drug that we put into the clinic should also have at least a monotherapy arm, and it should have a potential for single agent efficacy, because otherwise you are creating combination data, you're creating a signal, and you were mentioning investors, right? The investor will say, okay, that's a signal. Is it coming from the PD-1 or is it coming from your combination partner? Is it your drug? Show me the true data. And this is a phase 2 study where you have to run it head to head, and this is something we should avoid. Also, a big learning in IO specifically.
You fully agree that has been a general observation that monotherapy is essential. You look, however, at current therapies for, you know, breast cancer uses a chemotherapy combination of at least 5 drugs, and there's combinations are still there. So I think part of the question is clearly you need to show monotherapy efficacy of your immuno-oncology contributor. At some stage, it will become part of the therapeutic armamentarium. So part of the decision is when do you do that and how do you fund those studies? Should you wait until your product is registered and then doctors will start using it in combination? You generate phase 4 combination data. How do you fund that? Anybody has a comment on that?
So we have a small molecule which has really strong monotherapy data. So we have 80% cure rate in different kind of animal models. But the first question I receive is, show me combination data with classical checkpoint inhibition or whatever. So because, and if you are in the preclinical mode, what should I show if I have 80% cure rate? And if I, in a re-challenging experiment, I show that 100% of the mice completely destroy the tumor cells, what should I show with a combination? But it's always the first question we receive. Or can you show it with bites? Can you show the combination with ADCs? So there's only a slightly window, and no one is interested really on the effect of the monotherapy.
It also calls to the potential comparison to standard of care. You say you have to show standard of care, but sometimes the model is not relevant. Mm-hmm.
Yeah, it's also true that in the majority of clinical trials that we are running, I'm not in clinic, but of course I see what's happening in there. Are we really treating patients that are really monotherapy? Because these people are probably going through various phases of therapy, and sometimes we tend to say that this is monotherapy just because they have 2-3 weeks of no PD-1. Are they really monotherapy? So anyway, combination is in there anyway, and I agree with you, of course. That's the question that probably we've been asked so many times. What is the combination therapy for your clinical trial that is coming? So.
Yeah, so look, let me try to wrap this up and hopefully end on a positive note, right? So I mean, everybody in the room understand it is challenging. I mean, the biological innovation progress. I mean, if you just look into science, this is actually really amazing about all the data that are being generated. What we are talking here is a translational problem, translational in terms of development, in translation in terms of commercial, right? So that's the thing. So what we have heard today is you have to characterize your basic idea in an attractive way that it is unique and differentiated, right? While at the same time acknowledging, I think that's where Romana was going, basically, and I don't want to sound cynical, but mice models and PDX models have their limitations, right? The only thing you could say, actually, if it doesn't work anywhere, you probably would not proceed. But if it works in all the models, doesn't mean it works in clinical, right? So it is actually, it needs to have enough traction that people are convinced you should get it into clinics. If you end up in 3 years preclinical development, probably that's not the right thing. And in clinics, okay, now comes the whole thing. I think you need to have a plan up front. What is this smart demonstration of efficacy? You need to be smart in the execution, that you don't necessarily recruit some centers in Bulgaria with patients in the 12th line or something, that basically you can set yourself up from failure the way you design the program right from the beginning. And that requires what we were talking about, that you need to have not only scientific, you need to have pharma biotech expertise to really design your program right and cost effective, right? That's the whole thing. And third comes, that's then corporate development, right? So I think you need to go with the opportunity. If you can see that you run your trials in Spain cheaper, or maybe you find a paid partner that works with you developing your drug in China, I just go how you can win, right? Just sticking to the old paradigms and continue as we have always done over the last 20 years is not necessarily the recipe to success. So I hope that was positive enough, and I leave the last word to Esteban.
Thank you.
Finish on really on a positive note.
Absolutely positive. I hope you will share my enthusiasm for having had this discussion with this panel, showing that innovation actually affects in many different aspects of the value chain of biotech, from a discovery at university all the way through bringing really new therapies for diseases that currently have no good therapy. So thank you very much, and join me in thanking our panelists for a very good discussion.